All right. Good morning, everyone. We are so glad you're here. This is our fifth course in our swamp, freshwater, and ester and harmful algal bloom training series. Today, we're going to be diving deep into sample collection protocols and some things to consider when you're thinking about processing those samples or sending them to the lab for analysis. If it's your first time here, my name's Anna Holder. My pronouns are she, her. I work at the State Water Board and I am our Open Data Science Equity and Tribal Coordinator. And I'm going to be your MC today, as well as stepping in for one of our instructors. So bear with me. It's a new role for me today. Like I said, today we're diving into sample collection goodness. I think a lot of folks have been waiting for this content, so we're ready to go through it all with y'all. As usual, we'll do some logistical housekeeping, a Zoom poll. If it's your first time here, or you weren't able to attend previous trainings, don't worry, we got you covered. We'll do a recap of essential information that will give us all the essential grounding for today's training. And then we're going to dive right into sample collection details. Marissa and I are going to tag team this, and it's pretty heavy on information. So we're going to take a break, a 10-minute break, and then after we're done with all of our sample goodies, we'll do Q&A and do another break before we pass it off to our partners at the San Luis Rey Indian Water Authority, and they're going to talk about their lab and how they support tribes and all sorts of great stuff. We'll do Q&A for their awesome presentation and then do our wrap-up logistics as usual. So let's get to it. As you have probably seen, we're recording the instruction today so that we can post the recording online after the training for folks that registered or hear about it later and want to review after this moment. That said, if you have a question you want to ask, but you don't want it to be recorded, please let us know. Happy to pause the recording so we can have robust discussion as needed during those times. And then when you are ready to engage in that way, feel free to unmute yourself, turn on your camera if that's comfortable for you, or not, whatever works. Drop things in the chat. If I see something in the chat, I might elevate it verbally, but I won't say people's names. So if you want to engage anonymously that way for the recording, that's an option as well. I don't see anyone on the phone, so I'm going to skip the phone commands for now. Just a reminder, some of us use Zoom, some of us use Teams, some of us use other things. So if this is a new platform for you, a lot of your essential buttons are going to be at the bottom of your screen, your mute and unmute button, the chat, reactions, and that sort of thing. It looks like everyone's name in the participants list looks great, but if you want to rename yourself, add your affiliation, there's instructions on the slide on how to do that. We like to start these trainings with a Zoom poll just to make sure we're all awake. It helps me test myself too. So in a moment, I'm going to launch the poll. It'll pop up on your screen. We've got one question. We'll show that next. Select your response. These responses are anonymous. Press submit, and I'll share the results and we'll talk through them together. So for today, since we're really diving into sample collection, we wanted to get a sense of how experienced y'all are with collecting samples. These don't have to be harmful algal bloom related samples, water samples, bioassessment, sediment, air, whatever type of sampling you do in the field. We're just kind of getting a sense of how many times or how comfortable folks are with being in the field and collecting samples and things like that. So let me launch the poll. It should be coming up on your screen. And Based on your level of experience, our options are no experience, which is great. We're glad you're here. Hopefully we can get you ready to go in the field. Novice, so maybe you've helped others collect samples, but you're really following their lead and just kind of doing what they say. If I ever go out in the field with Marissa and team, I'm just hold the things that they tell me to hold, right? Because I'm not an advanced beginner or an expert. So an advanced beginner, maybe you've led the collection of some portion of the sample plan for that day, or you're an expert, you are managing and leading sampling teams. Okay, we've got 72% of folks have participated in the poll. I'll leave it open for another few seconds. Okay, over 80%. Amazing. And I think the other two, I know who you are and I know why you're not responding because you're part of our team. So I'm going to end the poll and share the results. And I'll talk through them together because you'll be able to see them on your Zoom screen, but they don't show up in the recording. So for the folks that are with us live today, we've got 44% say they have no experience with collecting samples in the field. Fantastic. We're so glad we're able to help you all get started and comfortable with doing that in the future. One person says they're a novice, so they've maybe collected some samples. And then we've got 44% of experts in the room. So loving that mix of experience, and hopefully we can learn from one another as well as we dive into our sampling methods that we use, but maybe you might use different methods that you want to share with the rest of the folks here. All right. Thanks for engaging in that poll with us. So as I mentioned, I am tapping in for Carly. Unfortunately, she's unable to join us today. So Marissa and I are going to tag team the swampy material today. And then our tribal highlight today, as I mentioned, will be with the San Luis Rey Indian Water Authority, who they have a lab down in Southern California, and they support the tribes with harmful algal bloom analysis and things like that. So we're gonna get to learn from them today, and we're really excited to hear about a tribally centered lab. Okay. This is where I need to pay close attention because I'm supposed to know a bunch of stuff here. So now we're just real briefly, we're going to do a quick recap of stuff we shared in one or more of our previous trainings that we think will help ground us all to jump into today's topics. So we're focusing on sample collection and a little bit of analysis today, which is foundational to any monitoring program, right? You go out to monitor and you need to collect your data. We've gone over this framework, the CCHAB Network Voluntary HAB Response Guidance, this 8-step process back in course 2. And we really went into depth into each of the steps of this 8-step framework. And this is the framework that our statewide harmful algal bloom program uses to protect, to respond to harmful algal blooms as we're made aware of them and to help protect public health. A critical step of this response process is monitoring in a couple of different places. And information collected by a monitoring program can be used to determine the appropriate advisory levels and corresponding signage to help folks near water bodies understand how to engage with that water safely. And then, inform the public of other safety precautions and things to be aware of. So in support of this monitoring, the Water Board has a pretty dense library of online resources available totally free on the internet, available to assist folks in visually identifying HABs, harmful algal blooms, both on the ground and by microscope. So this is a link to one of our visual guides, and there's a screenshot to one of our visual guides and a link to it on the bottom of the slide in that green bar. And for more information, we covered this topic on field screening and during course 1, where we covered it in a lot more detail. So to support sample collection for step 5, sampling for lab analysis, our program has developed standardized procedures for sampling for harmful algal blooms in freshwaters and saline lagoons found along the coast. And these standardized processes are essential to efficiently use staff time and resources to generate really good quality data and information and make well-informed response decisions. Again, these resources are available to you and anyone else on our FHAB portal, which is also linked here. And we're going to be linking to specific standard operating procedures or SOPs and other resources along the way today as well. The best monitoring can be a continuous process to be conducted when use of the waterways is expected. Sometimes this can be seasonal or it can be year-round. We'll talk a little bit more later about our pre-holiday assessments. As we know, folks in the summer months use waterways more frequently. The response process encourages surveillance prior to harmful algal bloom formation, just kind of like standard baseline monitoring, and continues once the HABs are present. And then it's important to monitor harmful algal bloom conditions over time, as they can be pretty dynamic and can become more severe before dissipating. So really understanding how the harmful algal bloom is moving through the waterways so we can continue to update our guidance accordingly. And then hopefully we're monitoring once everything's all clear and we can go through the process of deposting the advisories after the toxicity of HABs is below the public health advisory thresholds. And for those joining for the first time today, we've talked previously about why it's important to monitor for harmful algal blooms. And harmful algal blooms in California waterways can produce toxins that cause illness. Our program works with an interagency work group to track those illness reports across the state. And the graph on the slide shows, on the left, shows illness of humans, domestic pets, including dogs, and also fish and wildlife due to exposure of harmful algal blooms in water. And as you can see, the most illnesses that are reported to us occur during warmer summer months, which makes monitoring critical to inform advisories during those times. And then the map on the right displays cases of illness by county, demonstrating that this is an issue statewide. And, you know, as you can see on the graph on the left, kind of also year round with peaks during the summer seasons. So moving on from the response process, during our last training, when we talked about guidance to develop our monitoring plans, we talked about questions that folks might want to think about as you're developing those plans for yourself. And these are the questions that we use. So here is an overview of some general just water monitoring questions to assess water quality. And these assessments can include determining the status of water quality, trends over time, the pace of change, and then to prioritize areas of concern, and then importantly, determining causes of deterioration of water quality. To be more pointed and specific for harmful algal blooms, we've also included our program's monitoring questions. So these are our guiding questions for our programs, and we welcome folks to reuse them and remix them for your efforts. These were developed in 2021 as part of a strategic plan to develop a statewide water monitoring program to address harmful algal bloom concerns throughout California. This strategy had input from local and national experts to develop a holistic strategy for the entire state. And the strategy is catered to assess relevant scales like an individual waterway or an entire watershed or larger region. So it's also a useful tool when developing your monitoring goals and questions to address harmful algal blooms concerns, because you can kind of pick and choose your scale and the framework that you want to use. The strategy is centered around the assessment of status and trends of HABs occurring in waterways in California, to assessing the environmental drivers of HABs, incident response, as well as mitigation and prevention of harmful algal blooms. And then altogether, the information from monitoring informs these questions, those on the slide, to help prioritize actions and address the causes of impaired water quality. Our harmful algal bloom program at the water boards focuses on incident response for the protection of public health, since the water boards is the lead agency to address harmful algal bloom complaints. So by having a clear understanding as to where and what the percentage of water bodies that are experiencing HABs, we can redirect our limited resources to ensure public notifications are the most impactful. And we're still working towards implementing the full strategic vision and filling information gaps to develop predictive models, to do that year-round monitoring and appropriate mitigation and prevention measures. Once your monitoring questions and goals are established, then you get to think about the fun stuff, the monitoring. And there are different types of, we think about HAB monitoring in a couple of different buckets. The HAB program distinguishes these by resource intensity in terms of both how many people you have, your field staff, and then also sample and analysis costs. Again, our program focuses primarily on event response, as it's the lowest resource intensity and can be implemented on a larger spatial scale, which is important considering California is such a large and diverse state hydrologically and with the different HAB environments. The first type of HAB monitoring is event response monitoring, and that is triggered by a harmful algal bloom event, making this a reactive monitoring approach. So that's most in line with what we're doing here at the State Board. The second type is routine monitoring that provides the opportunity to better characterize when a bloom develops and the duration of the HAB. And it's often associated with an individual water body or a small group of water bodies. And those that do that type of monitoring can start to get a better understanding of the environmental context of when a bloom begins, the severity, its peaks, and this can help inform when to organize water recreation or water use events, ceremony events around proper conditions to prevent increased risk due to exposure to a harmful algal bloom. The last type of monitoring is ambient monitoring, and this is the most resource intensive, but it affords the opportunity to learn more about the other environmental stressors and water quality impacts to a water body. This data often can inform the status and trends of the waterway and future efforts to better manage a water body and identify actions to reduce the drivers that promote a harmful algal bloom in the first place. Ambient monitoring can focus on an individual water body or assess an entire watershed, and this is a comprehensive strategy to make improvements to at-risk or degraded areas and protect areas of high-quality waters. In the last training, we covered a couple things to consider when developing monitoring plans, and we shared this slide and spoke about the different types of monitoring that could be done according to low, medium, and high resource intensities. But we kind of did a broad overview of these topics. Today, we're going to dive into each one of these in much greater detail and provide information on sample collection and analysis procedures folks might want to integrate into their monitoring plans. Give you a menu of options that you can choose from. As we mentioned in previous trainings, not all cyanobacteria blooms produce toxins. So for those that have the ability, the toxicity ebbs and flows over time. So for monitoring goals, Determining the initial health risk is sufficient to inform heightened advisories, and for other goals, routine retesting is necessary to inform health risk over time. Okay, I think that's all for me for now. So now I'm going to pass it over to Marissa, who's going to share her screen and help us dive into the first details of our course.
Hi, folks. Yeah, I'm Marissa Van Dyke.
Oh,
I'm really
your
happy.
screen stopped sharing. Sorry to
Oh,
interrupt.
let me correct this. Let's get
No worries. I can cover
here.
the overview of this slide while you get set up. So in the first courses, we provided information on HABs common to California waterways, particularly cyanobacteria blooms and their unique characteristics and associated toxin production. In this section, we're going to share some reminders of that information that are important to consider during the planning for monitoring in the field and sample collection, but then also give some just tips that people know as folks that are in the field, things to remember before you're packing your trucks up and heading out there. So with that, I'll pass it over to Marissa. The slides look great.
Yeah. All right. Thanks, Anna, for that. Sorry about that technical difficulty. So again, I'm Marissa Van Dyke, and I'm happy to be here with you today. Thank you for joining us for this training event. All right. So as we go through these slides, wanted to do a little reminder, as what Anna said, we are providing a lot of content. The green bar on the bottom oftentimes has a link to the guidance that we're referencing or other useful links. And many of these slides are developed as like reference cards for you to reference in the future in whole or in part. Consider these templates to integrate into your own needs as you develop your own monitoring plan. All right, so let's get into this material. So many of the cyanobacteria blooms we encounter in California are planktonic. That's what we call them in general, or specifically they are seen as like free-floating blooms, where you can see it easily at the surface of the water, causing some discoloration. So those are those planktonic blooms, and we refer to that term a lot in our program. So one thing that's unique about cyanobacteria is that they have the ability to be buoyant. They can rise themselves up and down the water column, particularly during the daytime, to reach the sunlight and to photosynthesize, basically to grow. So targeting certain times of the day is actually really necessary to make sure your monitoring is most useful. And then when toxins are produced by cyanobacteria, these concentrate these concentrate these toxin production in their cells, and they form, for us to be able to see it without like a microscope, they form large masses like colonies, scum, mats that then we can easily see without a microscope. And in that is where these toxins are concentrated. So when we do monitoring, we go to a site, we do some field reconnaissance at those sites to pick the area that has the most dense accumulation to characterize the worst case exposure so that we can collect a sample that may have the toxins if they're produced. And although blooms can appear very dense, sometimes ugly, quite odorous, that density doesn't always correlate well to having toxin production. But it is the most conservative approach to target the most dense material. So you're most likely to collect a sample that's representative of that potential toxin production. And then sadly, we can't visually estimate the toxin production, right? We can see them and the plume density, but we need to send samples to the laboratory to analyze for that toxin concentration. And a few other considerations about cyanobacteria, because they're quite complex. It's also important to note that while the toxins are concentrated in the cells, there's still toxins that are released in the water. They're found dissolved, floating in the waterway as they decay, as the cells are damaged. as they grow. This is a normal process. So you want to collect that surrounding water as well with the material, so the lab can measure what's in the water and with the cells. And so when you're doing the monitoring in particular, there's also some other measurements that we find really useful to support understanding the environmental context of the bloom, including common water quality indicators, like determining the biomass or how dense the bloom material is. This is often done by doing cell counts under the microscope of the cyanobacteria or doing general chlorophyll analysis for the pigments, so you can understand how much cyanobacteria and algae are present. We also identify the type of cyanobacteria. This is identifying to the genus or species. We also call this taxonomy of the cyanobacteria that's present. And we can use handheld tools to measure, water temperature, pH, that kind of thing. And altogether, these analyses are really useful to understand the cyanobacteria community, how hazardous it is, and understand a little bit more long-term, how you can then learn a little bit more about your water body and the bloom to then refine the costly lab analysis over time for your program. All right, a few additional logistics before we continue. So in the field, there's several different detection methods that we can use. So this ranges from visual observations, literally just observing visually with the naked eye. We can also target sample collection at specific sites, or again, use these handheld sensors or sensors that we deploy, leave over time to collect continuous data. We oftentimes use a combination of these just because it helps extend our resources a little bit more to do that follow-up monitoring over time. There's also methods that go beyond data collection in person. We can also leverage satellite imagery to detect cyanobacteria blooms in large waterways in California. So if you're interested in this, check out course 4 for those specific tools. Also, determining how frequent your monitoring is important. So how often you're going to go to that site, and then also what time of the week to coordinate with the laboratory to make sure your sample submission is appropriate. You're not going to get samples there when they're closed, that kind of thing. Also, so that you can get results according to the timeframes that you need as well for your program. And then the locations for sampling are really important and can change over time depending on circumstances in the field. So for areas that are concerned, like recreational beaches or for drinking water intakes, it's helpful to do some reconnaissance again to gauge where to collect the sample for the highest bloom density, because day-to-day conditions like wind and flow rates can push the bloom density at the water surface, and that's for planktonic blooms. And also in California, we have benthic blooms. We go into this in many of our previous courses. Benthic blooms grow attached to the bottom substrates of oftentimes the flowing channel, like stream and reservoir, streams and rivers, and they can also be found in reservoirs, particularly in the shallow shorelines. And so these are less dependent, of course, on wind because they're attached, but you can find them in different flow regimes as well. And then further, when you're on the field, there's many different sample types you can collect, which we'll go into, and the depth of sample. You want to make sure that you're targeting that worst case scenario, that dense bloom, and not inadvertently avoid that scenario, just to make sure that your sample collection is really representative of what's present and to effectively use your resources. We'll go into that. Okay, so we're going to segue into our monitoring resources. We like to start off with the health and safety supplies, which are really essential to protecting staff that monitor in the field. These are items recommended by the FHAB program here on the slides. We often make kits of these, put them in bins so they're easy to grab and go. And we provide a checklist that can be integrated into your own health and safety plan as well. And for the resources for monitoring in the field and sample collection, those really depend on the procedures that you're going to be implementing in the field, what you've integrated into your planning. As you can imagine with those different resource intensities, right, event response, um, routine monitoring, ambient monitoring, there's quite a range of necessary resources and tools. So we're presenting these supplies and like the cost for lab analysis with these icons here in these general categories. Again, a visual assessment, collecting an actual sample, doing identification of what is there, and then that lab analysis. And these icons are repeated, like we've shown before. And so there's, again, now going into that resource availability, we've got that low, medium, and high. And that's how we've categorized these to help walk us through these types of monitoring. And the low resource approach really focuses on visual monitoring and some qualitative tests to determine the presence or absence of cyanobacteria. So oftentimes there's lots of algae, there's aquatic weeds, there's lots of things that look green, and we want to determine are they harmful or not. Then medium resource, those are the sampling efforts where there's going to be analysis costs. You're going to be collecting a sample, lab submission. These analysis costs are lower than full toxin analysis, but still also something that needs to be delivered to a laboratory and very informative to tell you the potential toxin production and also understand the identification of cyanobacteria. Again, understanding that potential toxicity. And lastly, we have the high resource approach. This includes analysis to quantify how much toxins are produced by the HAB in that sample. And you can use the preceding information also to refine that in a tiered approach, which we'll walk you through that. And we have many tips as we go through this to highlight the supplies that are necessary to store those samples appropriately, make sure they're kept cool and delivered to the lab so you get the best quality of data. And also don't lose your samples as well. There's a lot of efforts that go in out in the field. Great, so we're gonna go again with low resource, starting from the beginning. Okay. So when folks are in the field, for visual monitoring, when we get into visual monitoring, we like to provide training that you can see both planktonic and benthic blooms at the same site or just one or the other. So it's important to have understanding of both. Again, planktonic are free floating in the water column, and benthic are found grown attached to bottom substrate. They're most hazardous, these benthics, when the mats are detached. They're accumulated on a shoreline, in a swimming hole, their surrounding area of water intake. So again, they're quite mobile as well. So it's important to have that trained eye to be able to understand how these look like. And as mentioned before, we have a host of visual guides that can help staff be trained to really train their eye on what to look for. Here's a snapshot of a 2-page visual guide. We personally have these laminated and have them in our field kits, so they're handy. And also there's a separate one for benthics, and it's really useful to have both of them with you because you don't know what you're going to encounter until you're out of your water body and understand a little bit more about its trends. And then for a really deep dive and to support training on this, we have a comprehensive field guide that helps even identify down to the genus or genera of the cyanobacteria communities you may find. Also examples of green algae that are not toxic, aquatic plants. And there's also some resources from the ITRC available here and a really helpful video as well. All right, so for visual assessments, this is something that requires the lowest amount of supplies, really minimal equipment, and you don't need to step in the water, so you don't need to bring your boots or waders. It's really focused on paperwork and a camera and knowing what your location is, so you can go back and repeat that visual monitoring. All right, so as part of the low resource category, we also have some qualitative tests. These are simple tests. Again, doesn't require lab analysis, which helps you, again, distinguish cyanobacteria from other green organisms that look quite similar. And these are not necessarily 100% accurate, but they're really a key part of your monitoring. And these tests include the stick test, a gloved hand test, and a jar test. And these were developed nationally in the FHAB community and processes that we routinely implement. So first off is the stick test. So you can grab any stick or you can use a forceps. You kind of dip and pinch the material. If it comes up out of the water and there's pieces hanging off, it's likely green algae. These things stay more intact, right? They're true algae. And then cyanobacteria are known to fall apart. They can easily break apart, and it's likely cyanobacteria. Just helps you distinguish what you're looking at. Know what you should sample. The second complementary test, what we use most often, we put on a glove and we pick up the material that we're seeing in the waterway. And we scoop it up in our hand or pinch off some of the material from a rock or a branch or on the shoreline. And you put it in your palm and you rub your finger into it and it easily breaks apart, doesn't hold its shape. It's likely cyanobacteria. If it's algae, it's like hair-like stringy, it's likely algae. The third test is a jar test. So you can use any old mason jar. You don't need to use a formal lab sampling container. And what's important is that it's clear. So you want to put some of that material that you're concerned about with some water, let it sit for some time, let it in a dark place, preferably, and then it's going to separate. And this is aimed at determining if you have planktonic cyanobacteria, because those will rise in the water sample, in the sample container. All right. There's one last test that you can do, and that focuses on field microscopic identification. So this is an optional approach. It's using some more supplies, more equipment, and more staff expertise. So if folks have experience using a microscope and want to learn about how to distinguish cyanobacteria from algae under the scope, it's really useful. You can get a lower cost field microscope that doesn't require, you need to put it on a lab bench. And you can look at the sample under there to understand what you're encountering in the field and trigger some next actions in your monitoring, such as sample collection for analysis or based off of health advisory levels. It can trigger in California then posting if you determine that you have a bloom of cyanobacteria. And this is also advantageous just because you have real-time data. You have to come back to the location to take extra actions. But there is some room for errors, particularly because this is like a rough screening method, but it's a useful tool to put in your low resource category. All right, so now let's take a deeper dive into the higher resource categories, starting with sample collection or lab analysis. So collection for these samples is really foundational to characterizing the bloom and the potential health risks. There's only some information that we can collect without taking those samples, as we went over. And so in the medium and high resource categories, we're going to need to store those samples carefully and transport them for analyses. And complementary to collection of the samples, we also record our observations and some other measurements on field data sheets. Again, this can include like water temperature, the oxygen in water for understanding, for aquatic life, and other visual observations. And the sample collection really, the guides here that are provided from the program provide procedures that can be incorporated into different types of monitoring plans depending on the goals that you're targeting, and include standardized procedures and also example field data sheets that you can use as a template or as a whole. All right, so as mentioned earlier, not all cyanobacteria act the same. So we're going to start off with planktonic blooms and those sample approaches. This is, again, a high-level overview of the sampling procedure that's linked here at the bottom. And with planktonic sample collection, we want to do oftentimes a surface water grab or a scum sample. So starting with that surface water grab, we can do that from a boat, from a dock, from the shoreline. This is where staff take an appropriate bottle, they submerge it downwards near the water surface, and in U-shaped motion, you want to just pull it out of the water so that you can grab some of the underlying water and that discoloration and like the other particles that you can see visually into that bottle. You don't want to fill it completely just in case your really chilled cooler that you're going to place it into accidentally freezes the sample. So you want to break the glass because glass bottles are really important to use for cyanobacteria sampling. And then for scum samples, this is when you see scum. So sometimes we call it foam or like a dense film. But this is just a separate approach, and you want to choose that over a surface water grab just to use your resources efficiently. And rather than a U-shaped motion, you're going to skim the water surface, aiming at capturing mostly just that surface layer and some of the underlying water, just to keep that moist. So again, you don't want to do the U-shaped motion. And when in doubt, just collect the most dense looking material. And this is just a reminder that water surface grabs are done really quickly, and they can be done at the shore. If it's hard to reach, there's also sampling poles and other tools that can be used to sample from a bridge or a dock, including some submerged sampling devices as well that are available of use in our procedures as well. And this is just a closer look at that surface scum sample. And this is, there's additional processes in our procedure if your scum layer is exceptionally dense. There's a lot of layers. If it looks really discolored, kind of grayish, white, or brown, then there's a process that's recommended to mix the surface before you collect the samples so you can get the fresh material for lab analysis. Now, as we already alluded to, there's quite a bit of documentation that goes along with that, with those samples to understand what your observations were and help interpret the lab analysis results as well. This is an example of how our field data sheet looks like, one for each site. It's one page. And we keep a book of these just so that they're handy. And now moving on to benthic samples, sample collection. So again, this is cyanobacteria that forms mats or dense, dense kind of algal-like material that can become detached and found oftentimes on the shoreline as well, or floating down the waterway as you're walking around in the water and surveying your area. So again, cyanobacteria produce toxin within the cells, so we need to collect that mat material. We're not less focused on collecting also the surrounding water. And with how cyanobacteria mats are formed, there's quite a diversity. So you want to be able to survey the entire population best that you can. We'll go into that process in a moment. And benthic sample collection is particularly important when it's present because dogs and children are often drawn to this material. Dogs seek it out because of the way it smells. Children sometimes like to play with it, particularly when it's in the shoreline, when they're just kind of waiting and playing with it. So if you want to have a little bit more background on benthic mats, please check out course number 3. All right, so for the sample collection, we recommend a composite sample. This means collecting multiple pieces of the mat material in your area of interest and putting them all in one sample bottle. This is an economical approach to have a single sample, again, just like planktonic sampling. You have one sample per site to help you characterize. So it helps streamline things, particularly in your interpretation of what's going on. And this approach can be implemented in a stream or river, or also in a shallow shoreline on the lake. And sometimes for some monitoring purposes, you're interested in each different type of mat, so visually distinct mats. So you're going to be just collecting one mat sample at a time and matching that with like a photo to understand a little bit more about the broad range of benthic mats that you have in a site. And that's covered in a different procedure. But for health risk scenarios, we recommend composite sample. This is a snapshot of that benthic sampling procedure. It's a little bit more time intensive and a little bit more of a survey, a field survey than for planktonic sampling. This was developed, updated in 2025 based off of current science, and it involves assessing a 150-foot stretch of your area of interest. So staff are going to perform a coverage assessment. They're going to get in the water and collect a sample. There's also a breakout piece of the procedure if staff are not able to get in the water to also do this assessment from the shore, just making this very accessible for folks to understand the health risk for benthics, benthic blooms. And there's a different field data sheet that collects a few more different characteristics that you can find visually in that area. And when staff are surveying the area, they're looking for hotspots to target. And this guide helps walk through those main points of what you're recording on the field data sheet, how to do that spatial coverage assessment that can inform advisories, and how to collect that composite sample, where they're going to be taking pinches by their hand and putting all that sample material in the individual container for lab analysis. And this is an example of that field data sheet. And it also has an image, grid to help understand that visual coverage, because you're going to be estimating how much is present in that area of interest. And that can also inform health advisory procedures as well in California without collecting that sample for lab analysis. So that sample collection is optional. And again, if you'd like to have a little bit more information on that, we have a detailed video and procedures to go through for training. All right, so that was a lot of details to cover those broad range of blooms that we encounter here in California. And this is a quick reference card to help you just know what to focus on depending on the hotspots that you're seeing and how to sample, collect one sample per area of interest and just have an economical approach. All right, so sample handling. This is really important. You want to make sure that when staff are going out in the field, those samples are not lost and they're getting to the lab, maintaining their integrity, so you've got good quality data. So we have, again, lots of tips as a reference card to circle back to and incorporate into your monitoring plan. And just want to highlight a few of these elements. First off is the sample container is really important. You don't want to use just any sample container that you've used for other water quality monitoring. We prefer to use glass materials, and if you need to use plastic to avoid breakage, there's one type of plastic. It's a little hard to find. It's called PETG. If you have some questions about that, please contact us. We've found a couple of vendors that make those available. But most often we defer to glass, so you want to use some cushion material along with your ice packs when shipping your samples to a laboratory, or even just storing them in the field, transporting them in your vehicle. You want to prevent breakage. And also to maintain the integrity, you want to eliminate sunlight getting to the sample. So you want to keep them dark space, so just putting them in a chilled cooler is an easy way to do that. And already having that cooler chilled is really helpful so that you can reduce the temperature of that sample that you collected as fast as possible. It can be quite warm in the summer when we're doing sampling, and it's even more essential to cool your samples so that you can preserve that integrity and prevent any degradation of those cyanobacteria cells for identification. It's going to be really hard to see them. You're going to lose DNA, and you can also lose toxins as well if you don't preserve these samples appropriately. And also for transport to the samples, this should be prompt. So wherever you're going to be taking them, you want to ship them overnight when possible, the same day or the next day after sampling. And if you need to store them over the weekend, please refer to our procedures for some holding time recommendations, just to make sure you keep that integrity. You don't lose your samples. All right, and this is another reference card to really hit home the need to pack those glass bottles carefully and with lots of ice packs. We recommend not using crushed ice because that can melt and then leak, and a lot of couriers stop their shipping process if they see wet boxes or coolers. And also the labs will be measuring the temperature of your samples when they're received. And they need to see that they're below 10 degrees upon receipt. So that's about 50 degrees Fahrenheit. And so that's pretty low. And just to make sure that your costs for analysis are most effective, some labs will pause lab analysis and confer with you before proceeding or just stop entirely. So it's really important to keep these cool and to ship overnight when you do need to ship them to a laboratory. You can't transport them yourselves. All right, so here's some of the checklists to refer to for sampling equipment and supplies. This is the basic checklist that's recommended in our procedures. Much of what we spoke about, including cleaning materials, in addition to the health and safety materials as well. And then depending on the resource category of the methods that you're implementing in the field, this is a comprehensive list for the low resource separate from the medium and high resource to refer to understand what supplies you should plan for monitoring. And again, this really depends on your monitoring goals, what kind of sampling is necessary, particularly at surface or at depth, and what devices that's necessary to be most effective. Okay, so we've come to the end. We're going to take a break, but I'll hand it off to you, Anna, if you have anything further to add.
Yeah, we were literally a couple minutes early. That was a lot of material to go through, but wondering if there are any burning questions from folks that we can maybe tackle now before the longer Q&A period, or we can just come on back at 10:05, whatever, whatever you want, but pausing to see if folks have questions.
Yeah, I'll return back to that specialized equipment list if you just want to take a closer look at that and form any feedback that you have.
Okay. Not hearing any questions. I'm going to pause the recording. Please stand up, stretch a little, shake your brains. There's a lot going in them right now. And we'll see you back here at 10:05, a little over 10 minutes. When you get back from your break, give us a reaction to let us know that you're back and ready to go. Or say hi in the chat. Someone went sampling on the break. Wow. Talk about multitasking. Amazing.
That is amazing. Nice work.
That's intense. Awesome. All right. Well, we're going to keep on keeping on. So Marissa, take it away for our second part of sample collection.
All right. All right, so and welcome back, folks. My name is Marissa again. So I'm going to keep on digging through sample collection and monitoring tools with that detailed with the detailed sample collection processes that we already went over. We went over a lot of ground, but we have quite a bit more to go through. We have the medium resource and the high resource categories to go through. Then we'll bring it all together with the lab analysis considerations that bring them all together into a tiered approach for analysis that's really quite popular in many programs, not just in our state and in others as well, that can be useful depending on your monitoring goals in your planning process. All right. So In the medium category, we're going to get into DNA-based testing. And this may be new for many folks, or you've already used it in other monitoring approaches that you use outside of FHABs. So the first DNA-based test that we're going to go over is the DNA-based test that leverages qPCR technology, and that stands for Quantitative Polymerase Chain Reaction. We don't necessarily always speak this out loud, the whole terminology. We just call it qPCR for short. It's technology that's comes from the medical applications. It started being used in environmental monitoring since the late 2000s, and then more recently, it's been applied to monitoring FHABs. So it's a long history. And this analysis is really helpful as it tells you the potential of toxicity of your cyanobacteria community that's you're finding in your waterway concern. So specifically with this technology, you can determine how many toxin producing gene copies the cyanobacteria sample contains. Or in other words, since all cyanobacteria species have the ability or the capability to produce those toxins, meaning they don't have the genes to produce the toxins, it tells you if the bloom has that potential, and then informs what next steps you can take or should take to understand the health risks. And qPCR tests are highly sensitive analyses. They're available for individual cyanotoxin classes, so for microsystems, anatoxin A, saxitoxins, they're individual analyses that look at the toxin genes that are known in cyanobacteria populations worldwide and nationally to identify their capabilities. This analysis is also available for golden algae. It's a harmful algae, a proper one, not the cyanobacteria. It's also useful in water monitoring as well. And so, as mentioned before, we can develop a tiered approach of lab analysis. So when you know what cyanobacteria or genera you have in your waterway, and if you know that they can produce toxins, perhaps microsystems as your focus, then you can reduce lab analyses, only request qPCR for microsystems, and then see if that community of cyanobacteria has those genes in what amount, and that can then inform your next toxin tests to be able to compare it to the health advisories, which are again based off the actual toxin concentration. But since toxin tests are most costly, it's helpful to know which toxins to have analyzed. So we'll again go through that process in a moment. So this is reference card for some of the lab logistics, like the timing that's available. It's really rapid. You can get the results the same day of sampling if you have access to this method. And also some of the terminology that's used in the lab results. So for you to be able to interpret that information and to inform what toxin analyses that you'd like to have. But again, it tells you, does the cyanobacteria genera, that population, have the ability to produce toxins, even more so than just from microscopy. Another type of DNA analysis you can have performed is metabarcoding. And so this is analysis performed on your sample again, small aliquot of that sample, where they sequence sections of DNA to determine what cyanobacteria, genera, or even the species levels in your sample. It leads similar results to microscopic identification. And it's more accurate because it relies on the DNA of the organism to identify rather than the human eye, which can have error, or sometimes cyanobacteria itself is challenging to identify because it's degraded some, or it's on a certain life stage where it's difficult to distinguish among other genera in like that family. So again, it's a substitute that's more specific than microscopy-based identification. This is sometimes slower to get the results just for an economical approach because you can batch a bunch of samples, like 20 to 30 samples in one run. And so then sometimes, if you want to batch that many samples, you may not want to wait, or sorry, then you would want to wait to later to get your results. So oftentimes these are applied in special studies or when you're sampling many sites across a water body or some adjacent waterways as well to really understand and characterize those blooms. And so Again, this is, genus and species related, and really specific to substitute for microscopy. Okay, so let's spend some more of our time in the high resource category so we can pull that all together. And the high resource category, we start off with the toxin analyses. So we can quantify the toxin for these different toxin classes. Again, microsystems, separate anatoxin, cylindrospermopsin, and saxitoxin. Those are the commonly found cyanobacteria toxins that we find in California of concern. And those also have some health-based advisory levels for recreation. There's also national levels for drinking water, and also some guidance values for aquatic life and so forth to refer to. So before you pick which toxin test, you want to consider what cyanobacteria genera you have and what toxins they're capable of producing. So again, with those preceding steps. Or you can go straight for it and analyze your samples directly for the toxin concentrations. So with that, you have two main categories of lab analyses to choose from for that toxin test. The most common lab method that's provided in commercial laboratories, academics, maybe even your own in-house lab. The most common lab method is called ELISA. It's ELISA, which uses antibody binding as a detection method, and it's considered a biological assay versus an analytical chemistry assay. So if you do other chemical analyses for your water quality monitoring, you may not have come across this method, but it's very useful for cyanotoxin testing. It's going to provide you units in micrograms of toxin per liter of water, or also parts per billion. Again, how we get units of chemical in water samples for other monitoring, like for pesticides. So consider when you're looking at toxin analysis, think of the toxin like any other chemical for analysis when you're speaking to a laboratory and debating which method to use. These ELISA analyses are performed by the lab, each individual for each cyanotoxin class. So for total microsystems, there's going to be one method performed with one result, a separate test, oftentimes concurrently, just to get you rapid results. Then they'll just run a separate test for cylindrospermopsin, the anatoxin, and then for saxitoxins. The second most common lab analysis method for cyanotoxin measurements is liquid chromatography coupled with mass spectrometry. This is for short LC-MS, MS. There's some methods that use just single mass spectrometry, but coupling that further provides more sensitive and more accurate analysis. This is your traditional analytical chemistry method, again, used for pesticides and many other chemicals and water quality tests. It's an advanced approach. It takes more costly instrumentation, more experienced staff to be able to run this LC-MS. And again, you're going to get your results reported in units of microgram of toxin per liter of water or parts per billion, just like ELISA. It's important to note then when you're comparing the strengths and weaknesses between these methods, one's a little faster and cheaper to run. That's ELISA, and LC-MS is more precise, and it can be more costly in laboratories, and your cost is passed to you. It's important for your monitoring plan, particularly 1 year at a time or so forth, to pick one method and stick with it, because it's very challenging. It's basically not recommended to compare toxin measurements from different methods. Unless you're in drinking water analysis, oftentimes there's ELISA is run along with LC-MS because one goes faster for rapid public health protection. All right, so another part of high resource category is also microscopic identification by the laboratory. This is a little bit more resource intensive and costly than the DNA analysis in the medium resource that can also get you the identification. And if your program has staff with a microscope handling experience and they are interested in in-depth training for this taxonomic identification, we have a training guide offered here, also with some other training resources from other organizations that are really useful to get that training. It's something that you want to refresh on an annual basis. Microscopy identification is not easy, but particularly to get to the genus level, especially species. So really just family or genus level is necessary for cyanobacteria. And what most is useful is knowing, do you have green algae in that sample predominantly? Is that dominant, or is it cyanobacteria? So if you've got dominant cyanobacteria, you want to take next actions. Then if you don't have that capacity internally, many laboratories across the country, many in the state, offer microscope identification. And also while they're looking at who's there, identifying them, they also enumerate or count how much is there. So that helps you get with that biomass and that density that cell counts. And with microscopy, lab microscopy, it's really useful. It's most commonly used, when you resample your site throughout the FHAB season or your monitoring period, really understand how that FHAB community is changing over time, over the season, each week it can even change. So it's really useful measurement to understand how it's changing and understand with the environmental context what could be driving those changes. So you can start understanding what kind of bloom you can expect in this waterway, and also the severity as it changes to understand any precautionary measures that should be taken. All right, so lab microscope ID, this is again a reference card of the advantages, disadvantages, again, some alternatives. And this is just really useful, so please take a look at it. All right, so why is this identification useful? Again, we're trying to take that down to the downstream analyses. The graph here on the right shows common cyanobacteria genera and the number of toxins that they can produce. So yeah, it's more than one toxin class for many genera. You're not just, in a community of cyanobacteria you identified, you've got 2 dominant genera, they're only going to produce microcystins. It's sometimes not the case. Sometimes that community can have the potential to produce more than one toxin. So it's really important to evaluate the changes of your cyanobacteria community over time to inform what analyses are necessary in this high resource category, again, for your routine monitoring, activity monitoring, and really understand those health risks and build that economical approach in your monitoring. All right, so we're going to go into these lab analysis considerations that pulls that together, is what I was alluding to. So going back to the foundational components that we've been presenting here, like what Hannah started with, this is that reference card for the low, medium, and high resource. And that sample collection for lab analyses is happening in your medium resource and high resource categories here highlighted. That means you're going to need the appropriate container, you're going to need to chill it, and you're going to need to transport it to keep it fresh. And this slide presents that tiered approach that I've been speaking to. This is widely used in the laboratory setting to help prioritize what other lab analyses you should couple after learning the microscopy results and further. This really helps you prioritize your funding, particularly when you're monitoring many sites on a waterway. If one site, doesn't have any cyanobacteria present, then you don't need to often do any more further testing. And you can put your resources dedicated to another site that does have these indicators at these different tiered or these steps. So the collected sample is first screened by the laboratory for cyanobacteria. When particularly in the field, if you weren't able to confirm that, the lab can do that with the microscope ID or DNA-based methods, and then that will inform the next step. If the lab does detect cyanobacteria, then they'll perform that qPCR to determine if the cyanobacteria have toxin-producing genes, and if they do, which toxin-producing genes are there. So then you can choose from the many different types of toxins to analyze for that are common in California. And then you proceed with the toxin testing. So this tiered approach really has strengths in being cost effective, but I do want to highlight that it's not without some level of potential error, as this is not a perfect system. cyanobacteria monitoring is complex, so this is just a strategy really to be comprehensive and, and conservative with your resources. It's particularly so that you can monitor as many sites as you need, and also multiple water bodies, particularly in a watershed, which are oftentimes influencing your sites of interest. And this tiered approach is something that the FHAB program has employed for over 6 years. So we're available to walk you through this stuff. Also, share with you some pros and cons that we've observed while implementing this tiered approach. And this is probably one of the most useful charts here. That's a snapshot here. This chart matches the identification of the cyanobacteria genera, again, by microscope or DNA, to their potential toxin production. And this is a key that's used in many monitoring programs. The FHAB program maintains this chart for freshwater cyanobacteria to our best ability. We update this every 5 years based off of current science. And we use this in our tiered approach. Many other organizations, also even internationally, maintain such charts as well. It's really useful. All right, so when engaging with your laboratory, there's several analyses that you can choose from, right? You've got your DNA-based approach, you have your ELISA or LCMS. And this is, again, based off your monitoring program's needs. What are your goals? So do you need to know the toxin concentrations? Do you just need to know the potential toxin concentration? That's really helpful to understand. How accurate or precise do you need that toxin measurement? Do you need LCMS analysis that costs a little bit more to really understand specifically what components of the cyanotoxin class are present? So let's look at microcystins, because that's the most popular. That's a class of toxins. Many of the LCMS methods can only measure up to 12 of those, whereas ELISA can broadly detect the microcystins with less sensitivity. It can be a little oversensitive too. So again, strengths and weaknesses. These are outlined here in this reference card. To understand a little bit more about costs, I think we're going to get some questions about that. So let's go there real quick. So the DNA-based methods are quite a bit lower. You could do all 4 toxin classes for about $250 per sample. And then when you get to the toxin tests, if you want to do all 4 toxin classes again, so that's equivalent from qPCR, then you could be spending around $500 to $700 per sample, depending on the laboratory, and how quick you need your results. So again, that's really helpful to understand. All right, again, another reference card. This is really important to understand if you have a monitoring program that requires using lab methods that are accredited under some lab accreditation entity. There's a national lab accreditation program, and there's also a state-specific one. California has a lab accreditation program. And this reference card is current as of April. So this presents, what's available. Accredited methods are most often required for drinking water uses when there's requirements for monitoring under a permit or so forth. But for recreational uses, non-accredited methods are most often selected. They're more popular, more widely available at commercial laboratories, academic labs, and also internally. There's a lot of resources and time that goes into developing a method that's available for accreditation, and then even for the lab to gain that accreditation and maintain it. So again, if you want to employ some more cutting edge science-based methods, oftentimes those are not at that stage of getting that accreditation yet. So take a look here for the accredited ones. All right, and to help you understand what laboratories are available, both academic and commercial, we try to maintain a laboratory list in our program. This identifies their capabilities, and again, it's a handy reference. It's not comprehensive of what's available on the web, so as we're contacted by labs, we try our best to add them to the lab list. So you have a tangible resource here. All right, so now we want to go into a little bit more of the laboratory logistics, and communication in the labs is really important to coordinate your sample submission and ensure you're going to get what information you need in the time frame that you're expecting. So some labs have rush analysis available, but sometimes 24 hours or 48 hours. Sometimes the standard turnaround time for your results after they get your sample, after it comes from the courier. Sometimes the results are only available after 5 business days. So that's some things to consider about for turnaround time. That's the terminology folks use. So further, beyond turnaround time, the slide has some frequently asked questions that we ask, that others that we provide guidance to frequently ask about laboratories and to laboratories, and to understand what their capabilities are. Particularly shipping logistics is something that really can change every year. So it's really something important to survey the lab that you choose and to maintain that. So shipping couriers, oftentimes FedEx and UPS, are most reliable to provide that overnight shipping. It's really essential to keep your samples cool and within that holding time. So something that you want to consider also, that's increased costs for your monitoring program to be able to fund that overnight shipping and the time frame to drop off your samples. So it makes it to the overnight shipping, particularly if you're shipping out of state. That's another constraint on your sampling. Your sampling plan for week to week or month to month. Oftentimes we limit our sampling to Monday through Wednesday, so then we can make sure that we can chill our samples and ship them in time to reach the lab during its open business days. Some other questions are also presented here. Again, frequently asked questions. Asking for cell lysis. Oh, sorry, that's on the previous slide right here on the bottom. Cell lysis is really important in learning if it's part of the sample costs, because again, that sample that you've collected contains particulates, right? Your colonies, the scum, the mass, and you want that to be broken open so you can get measurement of all the toxins that are in your sample, both what was in the water that was collected in your sample and in those particulates. And most laboratories, the standard approach, have that cell lysis. That's just standard that they will do before they measure for toxins. So it's something good to check with, especially for some research laboratories and such. They give you those options. And then you want to learn a little bit more about their capabilities if they have a detection limit that's low enough to understand if your results are within a health protective zone, right? For California, there's trigger levels for advisories down to less than 1 part per billion of some of these cytotoxins. So you want to learn if that measurement can reach down there. For nationally recommended health advisory levels for drinking water is even lower. That's helpful to understand. Also, do you want to know what the whole what's present in that whole toxin class, or just individuals? So for microcystins, again, that class can contain over 200 different congeners. LCMS is really good at measuring about a dozen. And LCMS, and that's an LCMS. And ELISA can give you an estimate of all of the cross-reactivity, that all of those microcystins that are present. It can be a little bit too sensitive. It can be a little bit more heightened than if you use LCMS. It's important to know, the strengths and weaknesses of what you're going to be getting in your results. Also for antitoxins, again, it's plural. There's multiple ones of concern here in California. It's important to know if that test can give you an idea of more than just antitoxin. And also, if you have really high toxin levels, you may need to have your sample rerun. You want to know if it's greater than 8 parts per billion or 10 for some of the health advisory levels that are recommended in the California guidance. All right. And I can keep on going on this section if you like. So we went through a lot of material. A lot of ground we covered here today. So again, you have any questions, feedback, please start plugging those away in the chat. We're going to get to questions soon. Um, and so that went through, that categorization of approaches, costs, of supplies, to kind of really walk you through how to design your monitoring plan at the beginning, or how to advance it if you need to. Um, so for some monitoring goals of programs, you want to do some additional research. Oftentimes, to really understand the status and trends and understand what the drivers of the HABs are to inform some prediction models, or even just mitigation. What are you going to do about these HABs that keep on reoccurring in your waterway, right? So there's additional tools that are really useful as well. So this is a separate category away from those what we just went through. And these again are written as reference cards. So let's look at some of those tools in the toolbox. In the slide deck that's provided, we also have some additional notes too in the PDF, I believe. So that'll be really useful. So the first one that we often get a lot of questions about, and we see being implemented in more and more programs, is using imaging instruments such as the flow cam. That's a technology, that's an instrument you can purchase, and a flow cytobot. And these instruments are used to identify cyanobacteria and the algae, so that phytoplankton community in your water. It can be done in a greater volume and a greater pace. These instruments can do the identification without a person standing next to it, or putting a slide under the microscope. This can run independent of the program. It uses software, computerization, that kind of thing. The most often that we're seeing in lots of programs, even Department of Water Resources, many drinking water districts, is the flow cam. So that's imaging flow cytometer, and that's an image here on the left that we provide here. And this is something that's compact. You can use in a lab setting. It sits on a bench top, and you can feed in many water samples to then slowly get it analyzed, and those results are then provided on the computer to reviewed when you're available and you have staff available. So that flow cytometer is used to detect the cyanobacteria versus other phytoplankton by taking small amounts of the water sample, letting it drop through and taking images of it. And it's comparing the image of what it's capturing in each droplet to a library of images to be able to determine what family or genus that organism belongs to. So you're going to know about your cyanobacteria, your algae, your diatoms, so forth. From what we understand, in like 6 minutes, you can run an entire sample and unattended. So, there's some constraints. It's the upfront costs are quite high. From what I understand, that the machines can run about $80,000 to get set up. You also need to purchase a reference library or be able to borrow one from perhaps an academic or another entity that has one for freshwater organisms. This was originally developed and then worked for the marine environment to understand red tides, that kind of that environment. So there's different organisms that we're concerned with in the freshwater, and also the estuary, right? The brackish waters as well. Many academics in the HAB field have these instruments, and they offer partnerships to run your samples as well. The second technology is the imaging flow cytobot by McLean Research Laboratories. This is essentially an in-situ automated submersible imaging tool. So this is something that you submerge and leave in for 6 months to a year in the waterway. It's connected to a water quality station or floating buoy. It needs to be maintained incrementally. These can be found also in our coastal waterways, in the coastal marine bloom monitoring programs. And we're less familiar with their use, but there's some really great videos on these to understand a little bit more about their capabilities. Something that we use in our program and many others in the state is passive samplers, meaning you don't need to actively sample for the toxins. You can walk away, you deploy this device in the waterway, the water flows through it, and then you go back and collect the device and have that analyzed by the lab to understand what toxins were present. We use something called SPAT. It stands for solid phase absorption tracking, toxin tracking. And this is a synthetic resin. It's just like powdery. It's put into a mesh bag, and you submerge it in your waterway. It's going to absorb the cyanotoxins and other chemicals that are dissolved in the water as it comes in contact with it. It's really simple to set up, to use, to deploy, really low cost per bag to even create these. They're used in all sorts of waterways, freshwater to marine. You can analyze for many different toxins with some strengths and weaknesses. Very useful for microsystems, saxitoxins, so forth. You can attach them to a dock. You can stick them in the ground, and then you want to retrieve them maximum every 10 days. So that's something to consider in your constraints. So these are really useful for surveillance, particularly for benthic blooms and flowing systems. So that you can understand if there are toxic mats in your system upstream. It's something that's going to be able to detect those dissolved toxins that are passing through at really low concentrations, right? It's going to collect that and give you an idea of what's going on in that system as you walk away those 10 days or less. Analysis of photosynthetic pigments, particularly chlorophyll, is really common. A lot of eutrophication kind of monitoring programs to understand that severity of eutrophication or nutrient pollution in waterways. Those programs measure chlorophyll in their water samples. So cyanobacteria and algae both contain chlorophyll in their cells. So it's a common pigment to analyze. Cyanobacteria have phycocyanin, so then you can differentiate between the two by measuring both, understanding those ratios. It can tell you a lot about the primary productivity occurring in the waterway and whether a harmful algal bloom is developing or already occurring. So oftentimes there's monitoring programs that are set up with chlorophyll-based trigger levels. So, if you can't monitor all year round with all the different tools, you're looking at chlorophyll at a certain level, then you switch to, maybe you have cyanobacteria, so then you start adding additional analyses. These pigments can be analyzed with handheld field instruments. There's a couple of these on the slide to give you an idea. They also can be attached to field sondes, and that's in the middle image. And there's some benefits that they're really easy to use. You can take them out there, you get your data right away. However, they're not as accurate as measuring pigments in a laboratory. And many programs, or even if it's under a permit program, requires the analysis to be done in a lab. And then you're going to be using liquid chromatography, like we talked about for toxins as well, or by benchtop fluorometers, again, performed in the laboratory. This is really useful measurement to be employed. It's also used to verify satellite imagery for detection of blooms. And there's some links to guides on the strengths and weaknesses of each of these different technologies to integrate into your programs. All right, so for toxin testing, we focus on laboratory tests because that's the most reliable and most reliable to understand your results based off of health advisory levels. There's also, though, some field-based toxin tests that are commercially available to screen for different cyanotoxins in your waters. So these tests can be done in the field with some constraints. They take less than an hour or a little over an hour to run. They cost about half to maybe 2/3 the cost of a lab-based toxin test. So they can be a little bit more economical approach depending on your monitoring goals. However, there's some disadvantages with them. They're mostly used for presence absence of a toxin. Some of these tests don't have detection levels low enough for many of the advisory levels. So it's just something for recreational and drinking water programs. And if there's some requirements on that, and different programs, oftentimes they're not well suited for that, but sometimes they are for screening approach again, or like a low resource tier. They can be integrated in there. Some programs have been able to successfully integrate it into their low resource tier to then trigger additional analyses. The FHAB program, we don't use them ourselves because of the health advisory levels that we're comparing our results to for event-based monitoring. But again, depending on your goals, these may suit you well. All right, so I went a little bit over time, apologies there, Anna. But everyone, we really welcome any questions that you have, any feedback, any discussions. So I'll hand it back to Anna. Thank you for your time.
Thanks, Marissa.
Mhm.
That's a lot to absorb. We went through a lot in a short time, as we do here. Any questions for Marissa? We do have some monitoring experts in the room. We presented the stuff that we use, that we think about. If you have any different perspectives, approaches that you want to share with this community, we'd love to hear them as well. So feel free to unmute, drop something in the chat, and I'll pause here for a minute. Otherwise, we can go to a break. And also, if you have a question, but you don't want to be recorded asking that question, let me know. I know where the pause button is and I'm ready. So.
Yeah, I'll just add, after you digest this material, you take a look at it again. If you have some more clarifying questions, you can reach out to us. You've met me, you met Anna. Carly Nelson is my counterpart in the FHAB program, and we are available to do one-on-one discussions to clarify this kind of material and help you understand like how to integrate into your monitoring program or how to start from scratch. We can help guide you, regardless of if you partner with us or not. We're happy to provide assistance.
Okay. I'll pause the recording and let's come back at 11. Let me switch this so we'll get a little over 15 minutes and we will pass things off to San Luis Rey Indian Water Authority. That will be their lab experts, so they can go into more detail about their lab and practices and things to consider and things like that. So pausing the recording, we'll see you back here at 11 a.m. Again, I hope you had a good break. You were able to get up and stretch. We're in the home stretch. So I am so excited to pass the mic over to the San Luis Rey Indian Water Authority. And I just want to do a special shout out and thanks to Jeremy Billy, the Chief Executive Officer. He and his team have been critical members of our Tribal Advisory Committee and have been helping us developing not just this course in the series, but all of them that you've seen thus far. And I just am so excited to hear more about your work. Our meetings are usually super quick, down to FHAB business. And so I'm gonna stop sharing and hopefully, Jeremy, you're ready to go. It looks like it canceled everything. So you'll need all that practice. We were so on it during the break and then whoosh. Anyway, feel free to reshare and take it away. The floor is yours.
Hello, everybody. My name is Jeremy Billy. I'm the CEO for San Luis Rey Indian Water Authority, and I'm here with some of our team members. And I'm going to introduce one of our board members, Geneva Laughlin.
Hello, everybody. My name is Geneva Laughlin. I'm from the La Jolla Band of the Senior Indians. I represent my tribe on the San Luis Rey Indian Water Authority as the secretary for the board. I've been a board member for about almost 30 years. I'm a second generation. My son, Ahmed Aguilar, was the third generation, and we were ready to bring on the fourth if we had to. So my parents, Robert and Florence Laughlin, were involved early on. This is a 50-year water settlement, 50 years. Yeah, long time, a long journey. But I'm so happy to be here listening to the presentations because we are, this is what we've been fighting for with our settlement and our implementation agreement, which covers these areas for our bands. specifically for La Jolla, as we are on the headway of top of the river. And so we want to make sure that the water that comes down the San Luis River is clean, quality water, not only for ourselves, but everybody else. Our tribe uses our water for traditional reasons, for cultural things, our plants, our basket material, our herbs, things that we use. And in addition, it's a big recreation, economic development for our tribe. It not only provides recreation, but it implies employment, which we're located a ways away from the nearest town. So this is a main, and we are a main employer for not only our own tribal people, but for other people in the area. And so the water and the quality of the water is so important to us. And I'm so happy that you'll see in our presentation, our staff has worked hard to be able to monitor the HABs. And unfortunately, 6 years ago, when we started this journey, as you'll hear about in this presentation, we're facing some of the same problems we faced initially 6 years ago. So for those of you involved with your own tribes, HABs or water quality, to be sure and continue the good work that you're doing in testing so that our people can enjoy safe and clean water. Thank you.
All right, so thank you, Geneva, and we'll go ahead and get started with our presentation. Let me share the screen. Okay, so hopefully you can see that okay.
It looks great.
Let's start. Okay, it's not moving forward, so maybe I got a wrong screen. We just try to work out all the technical difficulties, and here we are having the same one. So 1 second.
It always works so well during the break. And then when people are watching, everything gets all mucked up. But we got time.
Let me go on again. Okay, so
Looks good.
let's see. Here we go.
Awesome.
All right, so the San Luis Rey Indian Water Authority that consists of 5 tribes: La Jolla, Paula, Palma, Rincon, San Pasqual, and we are a federally chartered government agency and ratified by Public Law 101-675 in 1988. Okay, and then we have sorry, I'm still having technical issues. What I'm about, part of the 5 tribes, they consist of 2 delegates and 1 alternate from each tribe. And currently on our board of directors, we have 3 of the chairpersons sitting on our board. So it makes things a lot smoother when we have to implement things in an emergency fashion, such as the HABs when it was detected. Okay. Yeah, nothing's working correctly at this point. Let's see. I apologize.
You're good. It's how we know a human's doing this, not AI.
Yeah. Let me
Sometimes
let me just try
you
to
have to
do it.
click on the screen itself within Zoom and then try advancing.
Let me try sharing a different screen then. Again, my apologies. I'm gonna stop sharing. Okay. Our video.
What started the whole dispute was that when the reservations were created, there was impliedly reserved for them enough water from the local water to meet all of their needs. But at the same time, virtually within the next few years, the federal government gave the same water away again, this time to Escondido and to Vista Irrigation District for their use.
They just signed a contract to build a diversion dam. Where did they think we were going to get our water?
In 1914, the Secretary of the Interior entered into an agreement on behalf of the Rincon Indians, but without their consent or consideration, and he gave Escondido Mutual Water District all the water rights to the river. In June 1922, William G. Henshaw entered into a contract with the United States of America on behalf of the Rincon and Pala Indians to build a storage dam and reservoir diverting water for irrigation. Local Indians protested the dam's construction as detrimental to their water supply, but federal officials turned a deaf ear. In November of that same year, the San Diego County Water Company succeeded Henshaw as the owner of the dam and contracted with Escondido Mutual to convey Henshaw water through the Escondido Canal. In 1924, the water company contracted with Vista Irrigation Company to supply 12,000 acre-feet of water stored in Lake Henshaw to Vista. Also in 1924, the Federal Power Commission approved a 50-year license with Escondido Mutual. This license included the Escondido Canal, the Rincon and Bear Valley power plants, and Lake Wolford Reservoir. The protected rights of the tribes were never taken into consideration. By 1946, Vista had purchased all the stock of the San Diego Water Company and the ownership of Henshaw Dam and 43,000 acres of land in Warner's Basin. In the following years, the two water districts took all of the San Luis Rey's water except a small amount reserved for the Rincon Band. There were no provisions made for the other reservations along the river. Reports by various government agencies warned that the tribes, as a result of water shortages, were being pushed out of the thriving agricultural business. Water supply was the basic and salient problem for San Diego County reservations, concluded a special committee report to the California governor and state legislature in 1965. The drying up of the San Luis Rey River has removed valuable sources of water, and economic operations of any magnitude are precluded due to the lack of water supply. Neighboring non-Indian cattle ranchers, dairy farm operations, and agriculturists have access to sufficient water to carry on their activities. Their fertile green acres with gushing water are in stark contrast to the brown, brush-covered and dusty expansion of the reservation land. In 1969, the Indian band sued the city of Escondido and the Vista Irrigation District, charging that the United States law protecting Indian reservation water rights had been violated, and that the Secretary of the Interior exceeded his authority in reaching agreements that gave away water rights on behalf of the tribes.
First elected to Congress, I met with the Indian bands and learned the history of the San Luis Rey River water dispute that had been going on for years. And so I found that the Interior Department gave the water rights to the river to two different parties, and that led to a serious legal dispute. We're talking about 16,000 acre feet of water. That's a lot of water.
4 years after the Supreme Court's decision, with no judicial resolution in sight, Congress enacted a bill sponsored by Congressman Ron Packard, titled the San Luis Rey Indian Water Rights Settlement Act.
I determined that to go on with litigation, the lawyers were getting wealthy, but they were making no progress toward a settlement. I think educating the people is crucial. I think it's very important that they understand that this dispute has personal effects upon every person, not only in the bands and in the water districts and the people that are serviced by these two water districts, which is hundreds of thousands of people, they don't have any concept of what this dispute really means to them.
Okay, let's see if we can get the other items operational. So that was supposed to be in the slide presentation, but again, it didn't work out. Let me
It was.
okay.
I saw it. I'm your witness. It worked before.
Okay. Let me put it back up. And let's share the screen. I don't know if it's working again.
I see your slides. They look good.
Let's try it again. All right, it looks like it's working okay.
Mhm.
All right. The video. So we'll go past the video. All right, so this you'll see a map. I know it's a little bit hard to read, but we're located in northern San Diego County, and our office is housed on the Paula Reservation. This map shows the close vicinity of the 5 member tribes in relation to the release of water from Lake Henshaw. It's the water body all the way to your right of the screen. And as a result of the settlement, in addition to selling 16,000 acre feet of supplemental water to Vista and Escondido, there's water that is released from Lake Henshaw that flows through the La Jolla's campground and diverted to Escondido and Vista and to the Lake Wolford and to recharge the groundwater under the Rincon Reservation. La Jolla uses the water that flows through their campground for not only income received through recreation, but also for cultural uses as well. Escondido and Vista continue to be responsible for operating, maintaining, and repairing and replacing the facilities of the local water system. Okay, a couple of photos, one of Lake Henshaw to the left and a photo of the water flowing through the La Jolla campground on the right. And this is the photos without the HABs. In 2020, remote sensing data from the state's HABs portal indicated possible harmful algal blooms, prompting Vista to begin monitoring for the cyanobacteria and the cyanotoxins. By June and July of 2020, laboratory results confirmed elevated microcystins levels in water release from Lake Henshaw. For this graph, laboratory results show that in June 2020, the total microcystins concentration was 29.7 micrograms per liter, exceeding the HAB's recreational advisory threshold. In July 2020, concentrations increased to 435.7 micrograms per liter, peaking in August at 2,973, as you can see from the highest point on the graph. And these are a couple of photos. One of Lake Henshaw to the left, and a photo of the water flowing through the La Jolla campground to the right with HABs. So during 2020, the HABs event led to suspended releases, reduced campground revenue for La Jolla Band, and lowered groundwater levels for Rincon Band. San Luis Rey Indian Water Authority, representing the 5 bands, entered negotiations with Vista and Escondido to establish both short and long-term mitigation measures, providing release criteria in November 2020, requiring weekly toxin monitoring 2 consecutive weeks below thresholds, and immediate cessation of releases if toxins exceeded caution levels. Let's see. This is our timeline. We added our HABs consultant, Dr. David Karen, in 2021, resulted in our advanced knowledge of HABs. Also, to assist in our communication with Vista and Escondido, the board approved hiring Levon Peck as our consultant relating to the HABs during the October 6th, 2022 board meeting, and an agreement was formalized on November 12th, 2022. June 2023, total microresistance concentrations were above caution and warning levels. As a recommendation from Dr. Karen and through the persistence from the La Jolla ban, peroxide treatment was preferred over copper. Vista and Escondido's 2022 HAB management and mitigation plan differed mainly in treatment strategy, favoring copper, while San Luis Rey advocated for more proactive peroxide applications. Through ongoing quarterly consultations, the parties strengthened communication, formed a technical group, increased peroxide and lanthanum treatments, and supported Rincon's development and approval of their water quality standards. This was a back and forth process because copper is a cheaper solution, but as you can see from the chart, the days after treatment lingering before water could be released. And Vista and Escondido still had to learn the dosage and when to apply peroxide before it was efficient or effective. Also, water levels plays a factor when trying to determine the most effective dosage. Just because of the level of the lake, it was pretty hard to determine. So even though copper eliminated the HABs bloom, what it did is that they just couldn't release the water, and without releasing of water, that did affect the campground's recreational use and this water flowing down that canal. So these are images showing that the bloom on June 5th, 2023, and a few days after the copper treatment in the lake. This was an issue for La Jolla because even though the water looked clear, releases to La Jolla could not occur during Father's Day weekend at the campground because it took 13 days after treatment to meet the permit residual levels for Lake Henshaw. So through our communication was, though our communication was improving during the settlement and implementing parties consultation meeting on July 18, 2023, hosted by the La Jolla Reservation, a comment was made by the local entities that receiving HABs testing results would sometimes be delayed, and that is when our previous president, Bo Mazzetti, made the announcement of, what if we started a lab? That way, because of the vicinity, we could shorten the response time, and if there was a detect, there would be enough time to retest and give the recommendation to release water. Because if there was a detect, this would stop the releases until a negative test was proven. During that meeting, President Mazzetti advised me to schedule an executive committee meeting to discuss San Luis Rey building a lab. President Mazzetti also advised Rincon, our Rincon delegate, to CONSAS to show Rich Williamson, our engineering, planning, and engineering director, a potential space on the Rincon reservation to do an assessment for a temporary lab. On July 21st, 2023, the San Luis Rey Indian Water Authority Executive Committee approved a $20,000 budget to start the preparation of a HABs testing laboratory, investing in getting the laboratory prepared with the related modifications to the room, purchasing furniture and equipment. On February 1st, 2024, the lease agreement was finalized with Rincon, and on February 6th, 2024, the Personnel Committee approved the laboratory technician position description, and on March 1st, 2024, the board approved a consulting agreement with Lorena Villegas as the laboratory technician so that our consultant, Dr. Karen, and Ali Lee could facilitate HABs testing because time was of the essence. Once the board had time to properly schedule a full announcement for the laboratory technician position, they approved a formal solicitation during the June 27th, 2024 board meeting. Lorena was hired as a full-time laboratory technician on September 3rd, 2024. And as a result, you'll see the drastic reduction in turnaround. So the commercial lab is about a 5-day turnaround with shipping and processing. The IWA lab is a 24-hour because we have local reception and immediate processing. Allowing the ability to obtain rapid results allows us to immediately halt or authorize water releases, neutralizing threats before they spread downstream. Operational independence. We no longer need permission or validation from the external agencies to act. Our data allows us to exercise our sovereign rights to protect our waters immediately. Okay, so this shows some of our key successes and outcomes, policy wins. Shifted Lake Henshaw treatment to the tribal preferred method of peroxide and anthanum. Rincon water quality standards approved by EPA created federally legal standards for water entering the reservation. Again, because it was a back and forth conversation between the local entities with Vista and Escondido, seemed like they were trying to find that cheaper solution with copper, but they just couldn't find that item that honed in like peroxide, which we were able to add and then release almost immediately. It's restored stability, revenue stability returned to La Jolla, to the La Jolla campground due to the data-driven decisions making and preventing unnecessary closures, protecting communities' health. And then the sovereignty in action. The tribes are no longer passive recipients of water quality data. They are the active auditors and regulators of their own resources. The lab provides the technical backbone necessary to enforce legal agreements like the settlement agreement and Rincon's EPA-approved water quality standards. And we're protecting our rights with our science. Anatoxin A, and this is your section. Okay, well, right now I'm going to introduce it to Lorena. She'll go over the next series of slides.
Hello, I am Lorena Villegas. I am the laboratory analyst. First of all, we appreciate the opportunity to present the analytical services we offer, and not only for the 5 tribes that form the IWA, but now also for the general public. Our analytical services are dedicated to the precise detection of cyanotoxins and algal pigments, both of which represent significant threats to the public health and freshwater bodies. We provide total concentration analysis for 4 key cyanotoxins: microcystins, anatoxinates, cylindrospermopsins, and saxitoxins, using the ELISA method to ensure high sensitivity and accuracy. And for those less familiar with the technique, ELISA is a gold standard biochemical method that relies on the highly specific binding between antibodies and antigens. And by measuring the intensity of the resulting reaction, we can precisely quantify the concentration of the target toxin. To complement this toxin analysis, we determine chlorophyll A levels via fluorometry using EPA method 445, the nanoacidification method. And this one serves as a high-resolution proxy for total algal biomass, which is essential for the early detection of plume events. Everybody knows that quality begins the moment a sample is collected. And for the team responsible for monitoring water bodies, they need to define sampling type, quality, quantity, and locations, among other factors. So follow your sampling plan and SOPs carefully, while also meeting all laboratory requirements. For our laboratory, we require a minimum volume of 20 milliliters per toxin. Amber glass bottles with Teflon lined caps are highly recommended to prevent toxin absorption. However, PETG containers are acceptable to reduce the risk of breakage during transit. A critical point is preservation. For anatoxin A and saxitoxins, we strongly recommend preserving samples at a 1 to 10 dilution at the time of collection. These preservatives can be ordered directly from Abraxas, However, if samples arrive at our laboratory unpreserved within the per 24 hours of collection, our laboratory will perform immediate preservation to protect the chemical integrity of the sample. In contrast, samples collected from microsystems and cylinders spermopsin analysis do not require the additional of chemical preservatives. And regarding logistics, we recommend to use permanent markers to label each bottle with names that match the chain of custody form exactly, and also to clearly indicate which analyses are required for each sample, along with time, location, and date of collection. Samples should be maintained every between 4 and 10 degrees Celsius in a cooler with ice or cold gel pads. Shipments must be sent overnight within 5 days of collection and kept dark during transport to prevent degradation. Upon receipt, and this is very important, we apply a strict protocol. We verify that temperatures are below 10 degrees Celsius and cross-check every label with the chain of custody to eliminate discrepancies. Our laboratory reports the total concentration of each toxin by performing a cell lysis procedure on all samples. And validated reports will be transmitted within 4 business days of sample receipt for regular turnaround times, but we do also offer other turnaround time services. Moving on to our protocols for chlorophyll A, the focus remains on maintaining the highest sample integrity from the field to the bench. For this specific analysis, we require a minimum volume of 250 milliliters collected in amber glass or HDPE containers. Because chlorophyll A is highly sensitive to light and temperature, these samples must be kept dark and cold immediately following collection and throughout transport to prevent degradation. We require samples to be maintained between 4 and 10 degrees Celsius in a cooler with ice. Regarding logistics, the process mirrors our toxin protocols. Please ensure each bottle is labeled with permanent markers to match the chain of custody form exactly, including the name of the sample, location, time, and date of collection. And samples should be shipped overnight within 5 days of collection, with a scheduled arrival at 10:30 to ensure prompt processing. Our laboratory strictly adheres to the ELISA protocols established by the maker, in this case is Abraxis, Gold Standard Diagnostics, to ensure maximum reliability. For microsystems, we use the EPA-validated ADDA ELISA method 546, which allow us to detect over 250 toxin variants. And we apply the same level of rigor to anatoxin A, cylinders, spermopsin, and saxitoxins. And for chlorophyll A, as mentioned before, we employ the EPA 445 non-acidification method via fluorometry. Regarding our quality controls, we have implemented a multi-level QC system applied to every batch. We maintain a high calibration standard with an R-squared greater than 0.98. And this coefficient of determination value ensures a near perfect linear relationship between the equipment response and the actual toxin concentration. Additionally, we ensure precision with a coefficient of variation of less than 10% for standards and 15% for samples. And this low CV percentage is fundamental as it confirms the repeatability of our analysis, guaranteeing that results do not vary due to handling or instrumental errors. We validate the accuracy of each run using a secondary quality control standard and perform matrix spikes to ensure a recovery falls between 70 to 130%. We also run blanks, and those blanks must always be below the detection limit. In the table, you can see both the detection limit and the reporting limit, and these thresholds have been established by Abraxas. The detection limit, this is the sensitivity threshold. And this is the lowest level where the test can say with certainty that the toxin is present. On the other hand, the reporting limit, this is the quantification threshold. And this is the lowest level where the test can reliably measure the exact amount of the toxin. Our laboratory followed the reporting limits set by the kit maker, which begins at the lower standard concentration. And by reporting at the reporting limit instead of the detection limit, we ensure that results are not just estimates, but precise measurements with the test confirmed calibration range. This is a glimpse of our daily operations. Our daily operations are built on mandatory environmental controls and instrument validations, recording the storage unit temperatures daily and verifying equipment calibrations. Our strategic weekly workflow begins with sample receipt and preparation. Midweek, we conduct dedicated batch runs to maintain our competitive pricing, followed by a multi-level data review by the analyst, quality control, and the manager before any report is published. However, during critical bloom events or upon request, we activate a rapid response exception. By bypassing the standard weekly batch schedule, we move samples immediately to analysis upon receipt. And this local capability allows us to deliver results within 24 hours, sometimes less, empowering our partners to make immediate decisions. This is a little bit about our cost. And as you can see, our pricing is structured under a volume benefit model. Our rates decrease as the number of samples increases. For example, the microsystem test drops from $145.50 for a single sample to $135.50 for batches of 10 or more. And for chlorophyll A, we maintain a flat rate of $55 per sample. These prices are per individual tests and are all A+, covering everything from receipt to the final validated report. While we offer 4 days regular turnaround time, we also offer 2 days and next day expedited options for time critical projects. We also wanted to share how we compare to standard labs. Because we specialize in cyanotoxins, we can offer a clear cost advantage. For instance, standard labs charge $165 for a single test of anatoxin A. Our rate is $155.20, saving about 6% per sample. And for batches of 10 or more, our rate drops to $144 compared to their $148.50. So our goal is simple, to stretch your budget so you can test more frequently without sacrificing data quality. This is just a friendly reminder. Our laboratory is located in Valley Center. It served as a local option for Southern California. And this geographic proximity significantly reduces shipping transit time while minimizing sample stress and degradation. But nevertheless, thanks to expedited shipping services, we can receive samples from anywhere. And partnering with us is a simple 3-step process. Contact us, discuss your project needs and pricing, Second, submit your samples following our guidelines and a completed chain of custody. And third, receive your validated digital report within the agreed turnaround time. Finally, please remember that when choosing an analytical laboratory, you should consider these pillars of trust. Clear pricing with no hidden fees. Look for transparent rates and volume-based models that facilitate your budget planning. Turnaround times that meet your needs. Ensure the lab can deliver results within your project's required timelines, whether standard or expedited. Flexibility during boom events. It is vital to have the ability to scale analysis and activate rapid response protocols during HAB crisis. Rapid communication and technical support, and multi-level quality controls and validated methods. Trust labs that use EPA-approved or validated methods. That's all from me. Thank you again for this opportunity to present our laboratory. And now we are open to any questions you might have.
That was so great to hear about the history of your lab and your capabilities and how you're serving tribes locally and beyond now, it sounds like. And it was good to hear from your perspective, you know, the reminders of keep your samples cold, keep it dark, to make sure you get really high quality analyses back. So we didn't even plan for that. So it's good to just reiterate those important tips. Any questions for the San Luis Rey team?
So I have not a question, but a comment maybe. This is Dave Karen. Thank you for the invite to join these meetings. I'm sure some of my water board colleagues are wondering, what in the world is Dave Karen doing on these? I know a little bit about HABs, but it was great to hear what you all are providing for the tribes. This is really great. I'm here obviously as an invited guest from the Indian Water Authority, and thank you for that. As Jeremy mentioned, I was brought into this project about 5 years ago. At the time, it was just becoming known what the issues in Henshaw were with respect to microcystins and antitoxin and the troubles it was causing downstream when the water was released. The interaction with Vista, as you know, has been, as Jeremy mentioned, has been a long and protracted one, and it's working. But it is, I think it was very intelligent of the Indian Water Authority to bring somebody in so they could interact with Vista and their consultants from a position of knowledge rather than vulnerability. And I think that's been really helpful and important. And kudos to them for doing that. Also, I'd say kudos for taking on the work and energy and input resources to develop their own lab. They have an excellent lab now for analysis via ELISA or LISA methodology. The laboratory is excellent. A large part of that credit goes to Lorena, who's done a wonderful job. It provides the Indian Water Authority with independence, self-reliance, and building their own knowledge base, which is really essential if they're going to be able to make good decisions in conjunction and interaction with Vista Irrigation District about water quality coming out of Lake Henshaw. So I just wanted to throw my 2 cents in that I think this is a wonderful thing that they've done, and it is a tremendous resource for any tribal entity looking to have toxins run, or anybody who is looking to have toxins run. And I think we'll see this laboratory spin up big time in the future.
Thanks for that insight, Dr. Karen. It's good to see you and hear from you. Yeah, we when we were starting to assemble the tribal advisory committee, we were thinking about this series. This was before Jeremy and team had joined. And one of the things, another tribal advisory committee member said, are there any tribally led labs in the state? Like, I haven't heard of any. And then we heard back from Jeremy, and in fact, there are. So I don't know of any others in the state. I don't know, Marissa, if you have any other thoughts or questions now that we have the Lorena with us, the team with us.
Yeah, it's great to see everyone and the opportunity to hear more about your history and all in particulars about your monitoring and the laboratory. It's an incredible achievement to establish the laboratory. So really big applause. Thank you for undertaking this effort. I've been involved in the HAB space for some time, and so I started in the California space in 2014, and at that time there was a laboratory network across the West Coast. Many laboratories that aren't here anymore, and some that did develop from a smaller scale to the ones that are available today. So it's a challenging field historically, and the West Coast really needs more capacity with for laboratory analysis, particularly with the increasing number of waterways that are impacted by HABs and the recurring HABs, particularly lasting longer than they used to. We really need that lab analysis and rapid lab analysis, so it's appropriate data that you're reviewing. And yeah, we've heard from many partners, particularly in the CCM network, the community-driven network in California, focused on HABs. Many partners have been asking about how to establish a laboratory. There's been some trials, and I'm really grateful to see in your new laboratory and learn more. You can see the data that it's cultural. You can see,
and it is challenging. Some of our challenges that is because it's not regulated. There's no urgency from agencies that do have water bodies that they're not forced to test. So that's one of the challenges. And then since we do have our temporary lab on the Rincon Reservation, we are in process of having a new established home, and we'll be moving to San Pasqual, and then expanding our laboratory area so that we can process, just have a more comfortable area for our lab. And we do realize there's obviously more advanced equipment out there, more expensive equipment that could even give us faster response times, and we'll grow when that market grows. But for the time being, the rapid rates or the rapid response that Lorena talked about, it is highly effective for us, but just because when we talk about release, there is a release schedule that we work out with the local entities when they're going to release water. If they're just a little blip, or just maybe it's a tad over, it gives us time to really test again versus waiting a full week and missing that holiday weekend, or if there was a ceremony that they had to use that water. That's the reasoning, at least for us. And then commercial aspects, that's down the road. Again, when maybe there is more regulation that will be needed.
Fantastic.
Yeah, I just wanted to add, as you brought up, there are no or very limited requirements in the state. And recently at the CCM network, we heard from the regulatory division at the water boards that requirements are on the horizon, both for the drinking water regulatory program and for surface water. There's more information on the water board's website, so if you're interested, we can put that in additional resources. But they are on the horizon. We're expecting to hear see schedules of outreach events and stakeholder meetings this calendar year.
Yeah, that's good information. Brand new information for me. So if you want to drop those links into the chat, Marissa, while we wrap things up here, I can add them to the slides as well so people can reference them moving forward. Last call for questions for the San Luis Rey team. And then just for clarity, Lorena, you mentioned that you're serving the local tribes, but then also the public. So does that mean you could potentially serve tribal governments up from Northern California? Or are you sticking with local tribes? Just curious about
The
the scope
general
there.
public, yeah. Yeah, I mean, everybody, everyone interested, we want to provide services, yeah.
Awesome. That's what I thought, but I just wanted to confirm. Amazing. Well, thank you again, Jeremy, Lorena, and everyone else there for joining us today and sharing your story and your capacity. It's really inspiring, I think, and wishing the lab the very best as you grow. I am going to share my screen again, and we're almost at the end of today's session, believe it or not. Let me see. We've got just a reminder of additional resources to support your harmful algal bloom monitoring and analysis. If you've been in any of these trainings before, this is old news for you, but we like to keep the reminders coming just in case. We talked a lot about the sample collection and monitoring materials today. So the monitoring program link can take you to the standard operating procedures, field checklists, data sheets, videos of sample collection methods, all those good things. In past trainings, we've talked about deep dive of advisory communication resources and signage and what those mean and things like that, as well as public health guidance, as well as our FHAB reports map. So when folks from anywhere in California fill out our bloom reporting form and say, hey, there's this weird green stuff somewhere. Here's information about it. That information ultimately goes to our FHAB reports map, so you can kind of see what recent reports have been coming in for awareness as you go out and utilize water bodies near you. This is, again, just a more detailed list of the type of online resources that we have ready to go for folks as you're developing your own monitoring plans, getting out in the field. Everything is completely free, and we highly encourage folks to reuse and remix stuff that we've already invested time and resources in developing, but then customizing for your own needs for your tribes or tribal organizations. And then, as Marissa said previously, if you're, gearing things up, you haven't maybe sampled with FHABs before, you want some one-on-one guidance or recommendations or thought partner or whatever, feel free to reach out to us. We are happy to provide that technical expertise to help y'all build your own capacity. The goal for us is to protect and restore water quality throughout California. Everyone's got limited resources, so we're hopeful through this training series that by building the community, we can all do a little bit more together. Again, we have, really focused monitoring around holiday weekends because of that increase in activity around water bodies. It's hot, so folks want to go to the river or the lake, whatever. As well as our year-round response. And so this is the list of our pre-holiday assessments this year. If you haven't been involved yet, there's still some time for maybe Independence Day if you're really fast, Labor Day if not, and then otherwise, these holiday assessments have been going on for a number of years. So if you're just thinking about gearing things up and doing the training this year, just keep in mind, happy to have folks opt in for next year as well. And there's a new flyer that we have for this year, as well as a link to the CCHAB network email list, which is where we announce early on around, I think, March or April to get folks ready to go to engage and opt into these pre-holiday assessments. And depending on how folks want to engage, We can also utilize the partner monitoring program year round. And this program, again, coordinates monitoring with tribal and non-tribal partners. And we can provide, sampling kits. We can fund laboratory analyses depending on what your interests and needs are. And the goal here is to really develop that partnership network, a community of practice, and share any resources we have to support your goals to protect water quality. That's important to the tribes. Again, so there's a flyer and links and all sorts of goodies. And we mentioned the ITRC. They've got a lot of great monitoring resources that we shared previously. I'm barely remembering the FHAB acronym. So for the ITRC, if that's new to you, that's the Interstate Technology Regulatory Council. And we just wanted to flag some that they have a really great monitoring page. And there's some tables here that can help if you want another perspective on another way different organization thinks about monitoring and procedures and things like that. Marissa, did I miss anything about this site that you wanted to flag?
Yeah. So they have a lot of incredible videos, like videos like real life spaces that have HABs or before they have HABs to understand how this differed support training. And then they also have consolidated kind of a resource center, particularly running how to do microscope identification. And then they have, again, another resource center to inform folks on different mitigation management technology, methods, and like how and what circumstances they can be applicable to start, understanding a little bit more about what those options are to do something about perhaps. We heard today about chemical treatment and alum treatment. So those are some of those options in the resource center to learn more about.
Awesome. I knew there was more that I was missing. Thanks for filling in the gaps there, Marissa. And then before we close today, just a reminder that all of we've been saying like course 2, course 4, those are listed here in order, as well as links to the recordings of those courses and the slides that were shared. As usual, I'm going to try and turn this around in the next week. So if you're on this call or you registered for this course, you'll be the first to receive links to the recording and the slides. And then we'll get it posted on our webpage, which is linked at the bottom of this slide, and announcements sent out to the FHAB Network email list, as well as our California Water Board's Tribal Matters email list to really distribute that information. As mentioned earlier today, folks are monitoring now. So we're going to take a break with our courses, our instruction to give folks space to monitor, because that's what you're doing as tribal monitoring experts. But we want to pick things back up later this year. I'm going to try and send out like an availability preference block around July to get a sense of, days or time of day that folks would prefer to see the rest of these trainings later in the year. October, November timeframe. We're keeping it flexible right now as we identify our tribal instructors and things like that. We want to talk about data analysis and interpretation. So you've done the monitoring, you've partnered with a lab, you get your data back. Now what? How do you look at it? Things to keep in mind. How do you interpret those results from our perspective? And we'll share what we do with that. And then as well, we heard some mitigation methods during the San Luis Rey presentation earlier. We're gonna November, December have I think like a panel type framework instead of hearing from Marissa, Carly, and Anna for the majority. We really want to hear about different research and mitigation studies that folks have been up to, just to kind of shine a light on the emerging nature of HABs being, growing and developing, and we're learning more every day. So keep an eye out for those things. And I think, yeah, that's it. We'll add the links that Marissa shared in the chat to the slide after this one. Have a wonderful summer, and thanks for joining us today. And we'll see you back here, hopefully around October time. Thanks, folks.
Thank you. Take care.